Welcome to this BioRails introductory video. The goal of this video is just to quickly explain the purpose of BioRails and the modules which are available to meet that need. BioRails targets the drug discovery process from early hit finding all the way through into the candidate submission. It applies the design, make, test and analyze cycle, which is repeated many thousands of times to transform hits into candidates for submission. At the design phase, it provides a store of results that can be used to interpret and design new compounds with optimized properties for the therapeutic application your drug is aimed at. It provides data for structure-activity relationships, for artificial intelligence and modeling techniques to leverage in order to predict improved therapeutics that can be used for design of new drugs, which can be measured using the make, test and analyze phases. In the make phase, it supports the registration of newly synthesized molecules coming out of design, the samples themselves, and the logistics of providing those samples to the test site, whether that's internally or out with the CRO, and planning the activities of testing. Under the test phase, it supports automation of robotics and instrumentation to transform the sample, test it in the biological assays, and ultimately then with the analyze module, analyze the raw data coming off the instruments and apply quality control techniques to make sure they're statistically valid in order to capture those results to feed back into the design process. If we look at a typical drug discovery screening funnel, we will be testing many compounds in the early screening phases. These are plate-based assays, and they're run in high throughput with automation. And BioRails supports that test process and the logistics surrounding it. Down into lead optimization, again, many more tests, both in vitro and perhaps some early in vivo results as well, used to optimize the compounds for various different types of properties, including their ADMET or DMPK properties. So measuring things like absorption, distribution, metabolism, excretion, as well as early tox studies as well, getting them ready to for submission and then to move into development after the NDAs have been registered. A lot of early discovery is based off screening, so we're using plate-based testing methods where we need to store mother plates and daughter plates as well as samples of the individual compounds that are being synthesized, ship plates between your organization and your CROs, but also distribute plates around the organization and perhaps make plates up for the lab to be tested. BioRails supports all of those workflows, including automated plate prep with instrumentation, liquid handling robots and automated storage devices. So BioRails is a modular product platform. That means there's one piece of software called BioRails and then 4 different modules that can be used to achieve that workflow that I've just described. The first one is called BioRails PTO, which stands for Project Tracking and Optimization. It allows project teams to request the assays that will provide the data to clarify the properties of their molecules. Once those assays have been requested, then the assay scientists will receive demand for those assays to be run. They can optimize the delivery by coordinating the logistics, so that's the inventory, along with the testing resources in order to get those tests run. The actual data, raw data, is analyzed using the BioRails DM module, which stands for Data Management. So this allows you to manage your data and intellectual property as you run these assays. It provides you with a lab workflow for running biological tests, and it leverages the Morphit software, which are analytical templates to both drive instrumentation and also analyze raw data into quality controlled results, which can be shared with project teams for design work. The inventory and material modules are used to coordinate the supply of both consumables and test materials for the biological testing. So first of all, the materials module is used to both register or access the chemical compounds or test articles that are going to be tested to check their biological properties. It can also be used to register samples which are generated from those biological tests. So in the example of HTS screening, we're going to register the compounds in your chemical registration, and then it allows you to access those and request them in the PTO module, test them in the DM module. The inventory module then allows you to track those materials as physical samples within containers. So they can be in vials, tubes, racks, plates, and other types of container. Those containers are then stored across different locations, maybe distributed between CROs and your own sites, but across buildings, floors, laboratories, freezers, shelves, and positions on that shelf. This provides the whole workflow with access to the quantity and location of materials to test, or indeed the location of samples which have been generated from those assays as well. So those four modules work together in unison to help you to transition between the logistics, the testing, and the analysis and publication of results. The final module is called BioRails BioHub. This is a central data warehouse which centralizes all of the results being generated from biological testing at CROs internally, and centralize them together into a query optimized data model for analysis and design of new compounds. And you can leverage some of our partner tools such as Certara's D360, Optibrium StarDrop, and even Spotfire Decision Site to analyze the data within the warehouse, which links together the biological assay, the test results, and the test article themselves to provide you with support for modeling, AI analysis, as well as structure-activity relationships. All the tools your compound designers need to improve and optimize the properties of your drug. So in this way, all the modules of BioRails work together to drive forward the design, make, test, and analyze phases. BioRails PTO is used to generate the demand from project teams. The BioHub data is brought together to allow designers to design improved and optimized molecules targeting your therapeutic area. The inventory and materials module are there to register those test articles and then distribute them to the test site, whether they're in vials or plates. The DM and Morphit are used to drive instrumentation to create plates, to read data off of readers and analyze, normalize, and fit curves or whatever the assay might be in order to quality control check and validate the results, so that those results can be published to the warehouse, the BioHub, to feed the next round of design changes. And in this way, you can optimize the progress of your projects, track the results, and track the progress to generate new molecules with optimized properties. And that's a quick overview of all the BioRails modules.